Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, please, if you could take just a quick second to like, subscribe, and maybe share this with friends and family, it really, really helps support the Sand Science Experience and lets us keep digging into this cool research for you. Okay, so today we're doing a deep dive into something many of us will eventually face: skeletal muscle aging. Why do muscles seem to shrink and get weaker as we get older? It affects how we function, right?
Yeah.
And we're really going to zero in on the cell's powerhouses, the mitochondria, to try and figure out what's really driving that decline.
Yeah, it's a fundamental question. And our source today is, well, it's a really solid piece of work. It was published in Cell Reports Medicine. It's by Cephas and colleagues from 2025. What makes it stand out is the scope. They looked at a really wide range of people: 51 inactive men and 88 active men, aged from 20 all the way up to 93. That's quite a span.
That age range is so important, isn't it? Because that's kind of our mission for this deep dive, to sort out a really tangled issue. We want to
Mhm.
separate the effects of just, aging itself, the stuff that happens no matter what, from the huge impact of how active someone is day to day. We're trying to pinpoint the actual, core mechanism that controls how quickly our muscle health declines.
Exactly, because clinically, this whole thing leads to sarcopenia. That's the term for the age-related loss of muscle mass, strength, and function.
Yeah.
And it's not a small problem. It really hits quality of life hard.
Oh, absolutely. And the numbers are pretty stark, aren't they? I mean, sarcopenia, it affects maybe 14% of folks in their late 60s, but then boom, it climbs to over 50% once you're past 80. That's huge. And that leads straight to, well, less mobility, more falls, just general frailty. It's a big concern.
For sure. And historically, when scientists were looking for the why at the cellular level, mitochondria were always suspect number one. The, let's call it, traditional view was that aging muscles weaken because their mitochondria mess up in two main ways. One, they get worse at making energy, so reduced respiration. And two, they start leaking out more damaging molecules called reactive oxygen species, or ROS.
ROS. Those are the free radicals we often hear about, right? The ones that supposedly build up damage over time.
That's the ones. So the old dogma was basically old muscles equals weak because the energy factories are broken and leaky, causing damage.
Okay, makes sense as a theory. But this new study, by bringing physical activity into the picture so clearly, seems to really challenge that.
It really does. They do some very thorough work, extensive phenotyping, as they call it. So physical tests, like how far someone can walk in 6 minutes, body composition using scans like DXA, measuring thigh muscle, and crucially, they took actual muscle biopsies from the vastus lateralis, a big thigh muscle, to look directly at the mitochondria.
Right, getting right into the tissue. Now, we kind of know exercise helps generally. Did the study confirm that?
Oh yes, definitely. The active people, unsurprisingly, showed better physical performance as they aged compared to the inactive ones. They also generally had less body fat, better insulin sensitivity, better metabolic health overall. That part wasn't shocking.
Okay, so activity provides some protection. But the real surprise came when they looked inside the muscle cells, at the mitochondria themselves, especially in those active older men.
Exactly, because if that old dogma held true, even in active men, their mitochondrial energy production, their respiration should have tanked with age. But it didn't. When they measured it properly, using different energy sources, the respiratory capacity was basically unaltered across the entire lifespan in the active group.
Hang on, let me get that straight for everyone listening. If you stay active, your mitochondria's fundamental ability to burn fuel for energy, it pretty much stays intact, even into your 80s and 90s.
That's what the data strongly suggests.
Well,
It implies that the drop-off in respiratory capacity we often see in older populations might be much more about being inactive than about aging itself.
wow. Okay, so that's one pillar of the old theory looking shaky. Activity maintains energy capacity.
Right. Physical activity boosts mitochondrial numbers and efficiency throughout life. That's clear. But aging per se doesn't seem to wreck the energy-making machinery if you keep moving.
Okay, but what about the other pillar, the ROS, the free radical damage? If that's the main driver of aging damage, it should be going up in the older folks, especially the less active ones.
Well, this is where it gets really interesting. First, mitochondrial H2O2 emission, that's the specific ROS they measured it, was not affected by age. It didn't increase in the older men.
It didn't go up with age? Okay, that's already weird compared to the old theory.
But here's the real kicker. ROS emission was actually consistently higher in the physically active men compared to the inactive men at all ages.
Wait, what? The healthier, fitter, older guys had more ROS coming from their mitochondria.
That's what they found.
Okay, that's genuinely mind-blowing. Because if the people doing better functionally have higher ROS, then how can ROS overproduction be the main villain driving muscle aging? It just doesn't add up.
Precisely. It forces a major rethink. If it's not about a fundamental failure in making energy, and it's probably not about being overwhelmed by ROS damage, then what is the key vulnerability that comes with age?
So we need a new suspect. Where did the study point?
The focus shifts away from energy output and towards, let's call it stress management within the mitochondria, specifically how they handle calcium.
Calcium, right? That's crucial for muscle contraction, but also cell signaling, even cell death pathways, isn't it?
Absolutely critical. So the researchers measured something called mitochondrial calcium retention capacity, mCRC. Think of it as a buffer.
Yeah.
How much calcium can the mitochondrion soak up safely before things start to go wrong?
Okay, a calcium buffer.
And related to that is the mitochondrial permeability transition pore, or mPTP. This is like an emergency release valve. If calcium floods in and overwhelms that mCRC buffer, this pore can pop open, and that's generally bad news. It can lead to mitochondrial swelling, dysfunction, even trigger cell death.
Okay, so mCRC is the buffer, mPTP is the point of failure if the buffer overflows. So the key question is, did that buffer capacity, the mCRC, decrease with age, even in the active guys?
That's the absolute breakthrough finding of this paper, yes. The mCRC, that calcium buffering ability, was significantly lower in older individuals. And crucially, this decline happened pretty much regardless of whether the person was physically active or inactive.
Wow. So exercise helps in many ways, but it couldn't stop this particular aspect of mitochondrial aging, this weakening of the calcium buffer.
Apparently not. It seems to be a more fundamental intrinsic part of the aging process in muscle mitochondria.
Did they see, like, a point where it really started to drop off?
Yeah, they did. The decline really seemed to accelerate after about 60 years of age. It's relatively stable before that, then it takes a steeper dive.
And that timing, that lines up pretty well with when we see sarcopenia really start to take off in the population, doesn't it?
It lines up remarkably well, yes, which strongly suggests this isn't just some minor biochemical change. It's likely linked to the functional decline we see.
Okay, so you have to connect it, though. Did they show that people with lower mCRC, this weaker buffer, actually had weaker muscles or performed worse?
Yes, they made that connection very clearly. There was a strong positive correlation. So individuals with a higher mCRC, a better calcium buffer, tended to have more thigh muscle mass, stronger knee extension strength, and they did better on things like the 6-minute walk test and a step test.
Right, so it's clinically relevant. Lower calcium handling capacity equals poorer muscle function and less mass.
Exactly. It suggests the vulnerability isn't so much about energy production failing, it's about the mitochondrion becoming less resilient to internal stress, specifically calcium stress.
This really shifts the focus. Okay, let's talk about the systemic angle they explored. They looked at a biomarker called GDF-15.
Right, growth differentiation factor 15. It's sometimes called a mitokine or metabokine. Basically, it's a signal released by cells, including muscle cells, often in response to stress, particularly mitochondrial stress. And the study really positioned it as a potentially powerful marker of overall biological aging in humans.
And how well did it track with age in this study? Was it a clear signal?
Incredibly clear. Plasma levels of GDF-15 showed an exponential increase with age. The correlation was really strong. R equals 0.83, which is very high for this kind of physiological measure.
Exponentially. Wow. So it's not just going up a little bit each year, it's really accelerating in older age.
Precisely. It suggests a building systemic stress state as we get older.
And the crucial link, did higher GDF-15 levels connect back to this mitochondrial calcium problem?
Yes, that's the synthesis. They found that individuals with higher circulating GDF-15 also tended to have mitochondria with lower mCRC mitochondria that were more susceptible to that calcium-induced permeability transition.
Okay, so that ties it together nicely. Rising systemic stress marker, GDF-15, reflects this underlying age-related failure in mitochondrial calcium handling, low mCRC, and this happens somewhat independently of exercise.
That seems to be the picture emerging from this data.
This feels like a genuine paradigm shift then, doesn't it, for how we think about tackling muscle aging, because it suggests maybe we shouldn't be solely focused on, I don't know, boosting mitochondrial energy output or fighting ROS quite so hard.
That's the implication. While those things might have roles, this study really highlights altered calcium handling and the mPTP as potentially much more central to the aging process itself. It gives us a potentially more specific target. Maybe we need therapies that stabilize the mPTP or improve mCRC, rather than just trying to make old mitochondria act young in every single way.
Right, it's about shoring up the specific vulnerability, not a total system overhaul.
Perhaps helping the older mitochondria manage stress better might be more achievable and more effective.
Okay, so let's try and summarize the big takeaway here for you, the listener. Physical activity, absolutely crucial. Keep doing it. It helps maintain mitochondrial numbers, boosts their performance, keeps you functioning better for longer, no question. But it looks like even lifelong activity can't completely stop this specific age-related decline in how well mitochondria handle calcium, that mCRC value. And that failure seems to be a really key, perhaps unavoidable, part of the cellular aging clock in muscle, closely linked to weakness and sarcopenia.
Spot on. And maybe one final thought, something provocative for you to mull over. Think about what happens after that mPTP opens under stress. One consequence can be the release of mitochondrial DNA fragments out into the main part of the cell. Now, this stray mitochondrial DNA acts like an alarm signal. It can activate inflammatory pathways, things like this CGS-STING pathway, leading to inflammasome activation. And these inflammatory pathways are known to actively drive muscle breakdown or atrophy.
So the calcium handling failure isn't just a passive breakdown. Opening the mPTP actively triggers signals that tell the muscle to waste away through inflammation.
It provides a potential mechanistic link, from the specific mitochondrial vulnerability, poor calcium handling, to the actual activation of muscle atrophy programs. It connects the dots in a new way.
That's fascinating. It really points towards inflammation driven by mitochondrial stress as a key player downstream of this calcium issue. Lots to think about there.
Definitely a key area for future research.
Well, thank you for joining us on this deep dive into, well, the real nuts and bolts of muscle aging. Really fascinating stuff. And a final reminder, please do like and subscribe to the Sans Science Experience podcast. We appreciate your support, and we'll catch you next time for more cutting-edge science.